As we gather for this final session of the reproductive system male module and the introductory animal physiology course, let me first congratulate you on successfully reaching the culmination of this learning journey. Your dedication and curiosity throughout the course have been commendable. In today's lecture, we will build on the foundational knowledge from previous weeks to deepen our understanding of spermatogenesis, puberty, hormonal control, assisted reproductive technologies. This session holds particular significance as it integrates and reinforces concepts from the entire course, providing a comprehensive understanding of male reproductive physiology. As you know, no subject can be mastered in few weeks. However, our primary aim has been to sensitize you to the shore of animal physiology to help you see the possibilities and scope within this field. Now it's your turn to dive into the vast ocean of animal physiology and explore its deeper aspects. We hope the concepts you have learned will serve as a guiding light for your academic and professional explorations. This being our last lecture in both this module and this course, I want to sincerely thank you for your active participation and enthusiasm. The knowledge and insights you have gained here will undoubtedly be valuable in shaping your future in animal sciences. Let's dive into today's session and conclude this course on a high note. The spermatogenesis, last class, I gave you what is the definition of a spermatogenesis, right? Now, we'll enter into the events which involves in the spermatogenesis. Spermatogenesis is majorly divided into three events. First is the spermatocytogenesis, second is the meiosis, and the third is spermiogenesis. Cyto means it is cell, right? And what is spermio? Spermio means it is related with the spermiation process. We'll see that in detail. Now, spermatocytogenesis, there are two important functions are there. The first one is that the mitotic division of a type A spermatogonia will take place. Now, this type A spermatogonia will divides and forms another type A spermatogonia, and then one it will just go and store for the future multiplications as a stem cells or the resting cells, you can say, right? So to maintain the population of the stem cells, the type A spermatogonia. And then this type A spermatogonia which migrates through the blood testis barrier or the Sertoli cell barrier towards the abdominal compartment, because the spermatogonia is the one which is present at the basal compartment. And you know the blood testis barrier, how it is formed and how it divides the compartment into basal and abdominal compartment. Now the spermatogonia from the basal compartment need to move to the abdominal compartment with the changes. So the type A spermatogonia is formed that will try to migrate the blood testis barrier and move towards the abdominal compartment with the different structural changes. Then type A spermatogonia will undergo the mitosis and it forms the primary spermatocytes. Now these primary spermatocytes are 2 in number, where it enters the meiotic division. Of course, I have given the pictorial representation also how the spermatocytogenesis will takes place. You can see that. The next is that meiosis. So meiosis, it occurs mostly in oogenesis and the spermatogenesis process in the body, right? So this meiosis, what is this? Here the homologous chromosomes will pair up, and these chromosomes, the homologous chromosome which is paired, that exchanges the genetic materials between the chromosomes will takes place in the meiosis, where the first meiotic division, in this division it segregates into two daughter cells, that is the progeny cells, right? Two daughter cells which is of haploid in nature. Before it was diploid in nature in the mitotic divisions, which is in the spermatocytogenesis we have seen the first phase event. The second is the meiosis, where the diploid become the haploid, that is two secondary spermatocytes are formed. Now the secondary spermatocyte will divide further, that means the daughter cells, and then it forms the spermatids. Mostly the spermatids are round in shape, right? Then in bull, from one type A spermatogonium to spermatid formation, how many will be formed? Around 64 spermatids will be formed from one type A spermatogonium in bull. The next will be the spermiogenesis process. The spermiogenesis process, if you take here, the round spermatids, what is formed in the meiosis as a result of meiosis, that will undergo the changes of a spermatozoa. Spermatozoa means it is a elongated structure. Okay, so from round structure it will become a elongated structure. Okay, so this is the head and this is the tail part. Now this is the mid piece and this is the distal portion of the tail. Okay, so from the round spermatid, how the sperm will become an elongated structure. That means the progressive morphological changes will take place in the spermiogenesis process. Now, what are the features you can find in the spermiogenesis process is that first is that formation of the acrosome from the Golgi apparatus. So, from Golgi apparatus it initiates everything, right? So, here the proacrosomal granules from the Golgi apparatus, it will form a acrosomal granule by just aggregating these granules, the proacrosomal granules, and it will get formed as a acrosomal granule, right? So that is the formation of the acrosome. Then the condensation and the elongation of the nucleus will take place. Now, and the third is that formation of the flagellum, which is required forward movement of a spermatozoa, and then whatever the excess cytoplasm is present, that has to be shed off from the spermatozoa. So this all will leads to the round spermatid into a elongated spermatid. Okay, spermatozoa, right? So based on the features, you can call there are 4 phases are there. This is the first phase, this is the second phase, this is third phase, and this is fourth phase. The first phase is called as the Golgi phase, where the formation of the acrosomal granules will take place. So from how the acrosomal granules is formed, that is from the Golgi apparatus, the proacrosomal granules will be formed. These acrosomal granules will be come and adhere together and forms the acrosomal granules. So that will take place in the Golgi phase. The next is the cap phase. So cap phase means it will try to form a cap. So where this cap is formed? In the head portion. Now, how this cap it is formed? This is just because of the nuclear condensation. The nucleus get condensed. The genetic material get condensed here. The next is that the reshaping. Okay, that means it is trying to form the structure along with the similar to the structure of the head, right? The nucleus. So that is the reshaping. The nucleus get condensed and then the reshaping will take place, and then it will form like a double layer membranous sac. So that is how this is the, it will form a double layered membranous sac. So this is how it will be formed. So this is a cap phase. A cap is formed on the head. Then third is that acrosomal phase. In the acrosomal phase, the most important is that manchet formation. It is nothing but the microtubules, right? So the manchet formation will take place, the manchet, and then that will get converted into a chromatid body. Okay, and then the next important associated with the acrosomal phase is the annulus. Annulus is nothing but a ring-like structure in the midpiece you can find. So these are some of the important events you can find in the acrosomal phase. One is manchet formation, then chromatid body, and then annulus, right? Then the last one is that maturation phase, where in the maturation phase the residual body is formed. So here I have showing the acrosomal vesicles. This is the nucleus, here the centrioles, right? And here the manchet formation is there in the second one phase. Then third is that acrosomal phase. You can find this is a mitochondria, right? It is just moving. And then the last phase, that is the maturation phase, where this is the residual body, right? And then in the midpiece, the nucleus, and then the acrosomal formation. Then the next is that spermiation. In the spermiogenesis, you will find the word comes from the spermiation. Spermiation means it is nothing but the gradual release of the mature spermatid or spermatozoa to the lumen of the seminiferous tubules. So usually, how the seminiferous tubules, if you take, it will be like this. So it is a inside there is a lumen. Now there are lot of cells. Now this is the spermatogonia, you can say, and this will be the spermatozoa. Okay, now this is the lumen. Now once it is matured, it will be released into the lumen. This process is called as spermiation. That means the gradual release of the matured spermatid or the spermatozoa into the lumen of the seminiferous tubules is called as the spermiation process. And because of this only it is called as spermiogenesis, right? Then that means the genesis for the spermiation process to happen. Then the next important point you need to remember in the spermatogenesis is that spermiation cycle and then spermiogenic wave. Because of this reason only we can call this spermatogenesis is like a factory, okay, production unit. How? The spermatogenic cycle is nothing but the time taken for the reappearance of the same stage within a given segment of the tubule. Like, for example, this is the segment of a tubule, right? Now, if we take the cross section of this part, then how much time it will take for the appearance of, for example, this is stage 1. How much time it will take for the reappearance of the same first stage of the morphological changes. First stage is the cell when you will find. Okay, how much time it will take for the reappearance of the same stage in the given segment of the tubule. Now, I told you this is the seminiferous tubule. I have taken this part. Now, here I marked the cell in the first stage. Now, how much time it will take for the reappearance of the same stage in that particular segment of the seminiferous tubules. That is called as spermatogenic cycle. You must clear about the concept, okay? Then next is the spermatogenic wave. Spermatogenic wave means it is a sequential change. Like for example, here it is 1. That means here it is 2, 3, 4, 5, 6. Okay, like that it's so on. Like here also, this is 1 means, then this will be 12, then this will be 11, this will be 10, this will be 9, this will be 8. Okay, like that again it will become 1. So, it is the sequential change in a stage of a cycle along the length of the tubule. Here you see the length of the tubule is the keyword. Okay. In the length of the tubule, here first stage is there. This first stage will become second stage, third stage, fourth stage, fifth stage, sixth stage, and so on. Okay, till the final stage. So, in the length of the tubule, if you take how much time, I mean the sequential changes are taking place, this is called as wave. Whereas, if you take a cross section of a seminiferous tubule and then how much time it will take for the reappearance of the same stage, then it will be called as spermatogenic cycle. Okay, clear? So, wave and cycle, this makes the seminiferous tubules as a high throughput factory for sperm production because of this reason. Okay, this is a spermatogenic wave, number 1 and first stage, right? This is the length of the tubule. Now, let me give you the first example. The bull, it will have around 4.6 cycles total. 4.6 what cycle? Spermatogenic cycle. Okay. Now, around 14 days it will take for the wave. That means each stage to complete. So, total 14 days, right? Then, so 14 days into 4.6 cycles is equal to 64 days. Where we have seen 64 days before, in the earlier slide also, I showed you, right? Where? Here it is there. The bull from one type A spermatogonia, it will give rise to 64 spermatids. So, to form the 64 spermatids, it takes 14 days. Okay, in this 14 days, around 4.6 cycles will take place in a bull. So, total 14 into 4.6, you will get 64. So, 64 days is required, okay, the spermatozoa to be formed. Then in pig, it will be 9 days. In sheep, it is 10 days. Then horse and rat, it is 12 days. Then bull, it is 14 to 16 days. Then human, it is 16 days, okay. And then in all the animals, depending on the species, around 4 to 5 cycles, it will take place. So, as a result, if you multiply like earlier the bull, then in ram and bull, it will take 60 to 70 days to form a type A spermatogonia into a spermatozoa, and in boar, dog and stallion, it is around 50 to 60 days it will take. Okay. Then the next important topic is that puberty. So what is puberty? So you can define the puberty where the male or female, when they can able to release the gametes and to manifest the complete sexual behavior sequence, then you can call that animal attains the puberty. So it is a male or female animal where successfully release the gametes and it can able to exhibit its sexual behavior, then you can call that animal attain or achieved the puberty. Or you can say in other words that puberty is defined as the establishment of the human pituitary and gonadal axis. So because once this axis is established, that leads to the successful steroidogenesis as well as the gametogenesis to take place. Okay. So you can, other words, it is defined as an establishment of the hypothalamo pituitary and the gonadal axis for the definition of puberty. Now coming to the age of puberty is that mostly in animals it is influenced by the physical environment. Yes, because the females which are reared in the presence of male will attain the puberty very fast, right, compared with the female which is reared in the female flock. And then photo period is another important aspect, because when there is a long day, that means the animal will try to eat more. As a result, it can attain the puberty very fast, whereas if it is a short day, then the animal will take rest more time and then they spend time on the feeding behavior. So as a result, the photo period play a major role. Then the breed of dam, depending on that. So if it is a big breed, then obviously it will take more time. Then it is a small breed, then it will try to attain the puberty as an early stage, and then sire also. And then environmental temperature. Yes, environmental temperature play a major role in animal science, because it causes the environmental stress to the animal. As a result, the hypothalamo pituitary adrenal axis get activated. That means that causes the release of the stress hormones. So that's have a negative effect on the productional and the reproductional aspect. Then body weight gain and the growth rate before and after weaning is also an important factor to attain the age at puberty. And then in animals, the onset of puberty is based on the body weight than to the age. In humans, mostly it is related with the age, but in animals, mostly it is related with the body weight. When calf or any young animals, when it reaches around 30 to 40% weight of its adult weight, then that animal will be achieved the puberty. So that is why the onset of puberty is determined based on the body weight in the animals rather than the age. Here you can see the animals and then the puberty time. So in dogs it is around 6 to 12 months, then in cat it is 5 to 7 months, and boar it is 5 to 6 months, then bull it is 10 to 15 months, then stallion it is 18 to 24 months, then ram it is 6 to 10 months, buck it is 5 to 6 months, and mouse and rabbit it is 4 to 6 months, right? Then next is that sexual maturity. What is that sexual maturity? It is a stage usually comes after the puberty. So first is puberty. Puberty, you know, that is, it is a successful release of the gametes as well as the exhibition of the sexual behavior. Then the next stage is the maturity, the stage after puberty, where the age or stage when the animal can normally reproduce, and exhibition of the secondary sexual characteristics such as ejaculation or the ovulation process, etc. So this is called as sexual maturity. So it is not that only it exhibits the sexual behavior, but it also equally it is important to normally reproduce and to exhibit the secondary sexual characteristics also is very play an important role in the successful fertilization and the reproduction. Then coming to the sexual maturity of different animals is given in the table, you can see. Then the seminal plasma. The seminal plasma, it is a plasma, that means it's a fluid that provides an environment conducive to the survival of the sperm. Okay, wherever this plasma comes or the fluid comes, and that is meant for the to provide a successful extracellular environment for the particular cells, because it supplies all type of nutrients and the gaseous requirement for any cell like that. Likewise, in seminal plasma also, it provides an environment conducive for the survival of the sperm within the female reproductive tract. Now, this seminal plasma is rich in electrolytes, fructose, ascorbic acid, as well as other vitamins. Now, the fructose is one important compound, the chemical, which is required to provide the energy for the spermatozoa, okay, in the female tract for the onward progression. Then, what is the semen? Semen, usually it is used to collected and used for the breeding technologies such as artificial insemination and the in vitro fertilization. Mostly the semen only collected. What is that semen? Semen contains seminal plasma and then spermatozoa, that is sperm. Okay, so this is called as semen. Mostly these semens are collected and it is evaluated and for the breeding technologies such as artificial insemination and the in vitro fertilization. Then next will be the spermatozoa. So far we have seen what is spermatozoa. So spermatozoa consists of head and tail. Now tail contains the midpiece also, right? Then head usually contains the nucleus. Nucleus contains DNA and the acrosome. Then the tail portion, it is made up of microtubules as well as the microfilaments that provides the motility for the spermatozoa. This is the most important because this provides the motility. Many times there are certain abnormalities where the tailless head you will find. That means what? Then in that case, even though the acrosome is in good condition and then all the enzymes are present and the nucleus which contains the DNA, everything is present, but that spermatozoa cannot be used for fertilization because it cannot move, I mean in the reproduction part. Then the middle piece of the tail where it's made up of mitochondria that provides the energy for the motility. Okay, that is another as important aspect. Then acrosome, the acrosome which is present in the head contains the hydrolytic enzymes such as acrosin and the hyaluronidase, which is responsible for the acrosome reaction. Now, here you can see the ejaculated volume in ml of different domestic species, where the boar or in pig you can see around the 150 to 500 ml of volume of semen used to be released, right? And then ejaculated. Then sperm concentration, it is high in the ram as well as the cock. Here in this species, the concentration is very high, that means millions per ml. Why the concentration is very poor in the boar? Because here the volume is more, that's why the concentration is less. It is almost inversely proportional, you can say. Then motility of the sperm, you can find it is majorly in the cock as well as in the ram. Then morphologically normal sperm percentage, that is also almost higher in the cock, right? And almost all the species, right? It is 95%. And the next important is the fructose, which provides the energy for the spermatozoa around 460 to 600 millimoles. It is present higher in the bull than the other semen. And then sorbitol content is higher in the ram than the other species. Then inositol, that is around 380 to 630 milligram per 100 ml you can find in the boar. And then ergothioneine is another compound which is present only in the stallion, okay, in a higher concentration than the boar as well as the cock, whereas in other species it is not present. So ergothioneine is majorly are present in the stallions. So here you can see the comparative spermatozoa of different species. This is cock, this is rat, this is horse, ram, boar and bull. So you can see the different spermatozoa. This is the head portion and this is the mid portion and then this is the tail. Completely it is a tail, and here it is an annulus, that is a ring like structure, it is present. And then now coming to the acrosome reaction, how it happens, the fertilization, right? So these spermatozoa will come and try to make a contact with the ovum, right, where it releases an exposure of the enzymes during fertilization. Which enzymes? In last slide we have seen the acrosin as well as the hyaluronidase. Hyaluronidase, what it does, it try to release the cells from the basal membrane. So usually it is sticks, okay. Now it come and dissolves the sticky nature of that cell. So as a result, the cells will try to move, leave. So as a result, it creates a gap. So the same way here, what happens, the acrosome, the hyaluronidase and the acrosin will come and during fertilization, it will fuse with the acrosomal membrane with the sperm's plasma membrane, and that releases the acrosomal contents which pass through the zona and fuse with the plasma membrane of the egg, okay. So this is how the acrosomal reaction will take place. Next, coming to the hormonal controls. First, the GnRH from the hypothalamus that will go and act on the anterior pituitary where it releases the FSH and LH. This FSH and LH is called as the gonadotropins. So FSH and LH is the primary endocrine regulator of the testicular functions. FSH act on the seminiferous tubule, promotes spermatogenesis. That is, for initiation, FSH is required. Whereas LH, I told you, it is ICSH in the males, which act on the interstitial cells. That is why it is called the ICSH, promotes the secretion of the androgens or the testosterone, which is necessary for completion of the spermatogenesis. Now, the serum testosterone, whenever the concentration increases, it causes a negative feedback by stimulate. It will go and act on the hypothalamus that decreases the release of the GnRH. So, the testosterone have a direct effect on the anterior pituitary that suppresses the LH, increased testosterone. And the FSH, if you see, that will go and stimulates the sustentacular cells, the increased FSH, and that will releases the inhibin. So, this inhibin have a negative effect on the anterior pituitary where it suppresses the FSH itself. So, it is an auto cut mechanism. Okay. Then coming to the other functions of the testosterone apart from the spermatogenesis, it development and maintenance of the libido, that is a sexual drive, and then secretory activity of the accessory sex glands and the general body features such as secondary sexual characteristics, descent of the testis in the fetal growth. Only in the fetal growth it is the descent of the testis is the function of the testosterone, whereas in the other cases it is the after birth, that means once the parturition has taken place. So the same thing is given in the flow chart. Then coming to the breeding soundness evaluation is assessed in the semen based on the collected and to evaluate the fertility of the breeding bulls. Now here when we can say that a bull is really sound for a breeding purpose, when it is having a progressive motile spermatozoa, which is the best indicator for selecting the breeding bull. That should have almost more than 70% of the progressive motile spermatozoa, and it should not have more than 20% of the primary defects it should not have. Then in that case we can select that bull as a breeding bull. We can select that semen and then it will be used for the artificial insemination and other ART purposes. Coming to the soundness, there are different types. You can say one is structural soundness, reproductive anatomy based, and then semen quality. So structural based means it is body condition, general health, leg posture and other things. Then reproductive anatomy is scrotum, testicles and the testicle circumferences. Based on that you can assess the bull. And then semen quality, motility, morphology and foreign debris present in the semen. Based on that you can assess. Then coming to the abnormalities, there are three types of abnormalities. First is that the primary abnormality, which is development within the testis itself. Then second is the secondary abnormalities, which defects arise during the epididymal transit. Whatever the problems or the defects are arise, that will be classified into secondary abnormalities of the spermatozoa. Then third is the tertiary abnormalities, which is taken place during the handling or the processing of the spermatozoa. Based on that it is classified. And the last part is that assisted reproductive technologies. So this is the technology used for semen analysis, such as hypoosmotic sperm smelling test, then through the CASA and then fluorescence activated cell sorting. So through this ways we can analyze the semen, and that will be used for assisted reproductive technologies for different ways to produce the young ones. So sexed sorted semen is also, that is X and the Y chromosomes. Based on that it will be. So nowadays the X sorted semen which contains X, so that will give a female calf, right? Then ICSI, interstitial cell stimulating hormone, that 1 ohm requires only one spermatozoa for that. And then IVF, in vitro fertilization, then GIFT, gamete intrafallopian transfer, then ZIFT, zygote intrafallopian transfer. These are all comes under the IVF only. Okay, so the same thing is given, that is assisted reproductive technologies, some different methods. One is the fertility medications, that means some hormonal therapies will be given so that the fertility infertile problem can be rectified. Otherwise they will go for assisted reproductive technologies where the intrauterine inseminations or the in vitro fertilization will be done. So in in vitro fertilization, embryo transfer, ICSI, GIFT or ZIFT will be done. So today we have seen the spermatogenesis process, and then what is spermatocytogenesis, then meiosis in the spermatogenesis, and then spermiogenesis. Okay, then what is the puberty, and then the hormonal control and the assisted reproductive technology which involves in the male reproduction. So this ends our final lecture. Thank you for your patience learning.